So thank you so much, Ron and David, for the invitation to be here. It's an honor to be in this series. And I am really grateful to everyone who showed up here to do a bit of quid pro quo. I know you're not going to ask questions while I'm talking, but I'm going to try to keep this short so we have more time for discussion. So we'll see how it goes. I'll be as efficient as possible. And I wanted to highlight a few things that we have going on in Chicago to try to encourage all of you to think about Chicago for your next, sort of long stay or visit. We now have an NSF-funded Physics Frontier Center for Living Systems that's headed up by Margaret Gardell and Arvind Murugan at U Chicago. And we also have the NSF Simons National Institute for Theory in Mathematics and Biology that sits in the Hancock Building in downtown Chicago. And both of these, new centers and institutes have programs to bring folks in for convening activities. Basically, we hope you'll visit us. The third coast of the US in Chicago is a wonderful place to be. I wanted to start out by thanking the folks that I will be sharing part of their work, part of their thesis work, part of the work that got them their current jobs at other universities here. And these are they. This is the whole group right here. And in particular, Caroline Holmes and Ben Hoschel just graduated less than a year ago, and I'll present some of what they've done. Chris Lin, Wave Ngam Predicorn, and Sue Wang are all postdocs from the group who in January almost simultaneously started their own faculty jobs. Chris at Yale, Wave at U Sydney, and Sue at SUNY Stony Brook. So if you are looking for a postdoc, please check these folks out and generally keep an eye on these spaces. Okay, so as David in the lovely introduction gave, the introduction is also this music vignette I felt is a lot of pressure. This is beautiful music, like with this crescendo and I hope to hold your attention to the level that preamble did. And we ended with like Jacqueline Dupré about like blow our minds with the next note and then it cut. So hopefully I'll be able to deliver on what you have, in your soul right now from that. So, all right. David gave a lovely introduction and talked about optimality. And I think about this in terms of what matters to a biological processor. And I think we should and could have a nice discussion about whether anything in the brain is actually optimal, because our notions of what matters to the system might not align with what the system cares about. And we're not here to discover that biological systems are sort of optimal physical or engineering devices, though they might be in some sense. We have to think about this in the context of natural selection and what matters to the organism. So when I say biological processor, I mean that the output end goals of that little computer that's written in biological stuff has to be the behavioral goal, has to suit the behavioral goals of that organism. So in the places where we find optimality, we have to think about why we've discovered that. And most of the time, we've hit on a place where that biological system, that fly, large monopolar cell, that piece of the retina has been sculpted over evolutionary time through competition with conspecifics and other organisms to do that job very, very well. It's not that biological systems just have efficient, sort of optimal processing as their like sort of built-in cost function. So I want to try to grapple with that. And to do that with you today, and to do that, we've employed some tools from machine learning to get that job done, to steep ourselves, to embrace the complexity of real biological behavior. It is, as a theoretical physicist by training, it's terrifying sometimes, but very rewarding once you find the places where you can really dig in and make contact with what organisms themselves care about. Okay, so let's go through a few more of these thoughts. So optimal coding, for us, that's going to be maximizing information transmission subject to constraints. Usually the constraints in any paper that you read or things that you hear about are buried. The constraints for us are everything. the constraints are where it's at, and particularly the constraints are the behavioral goals of the organism, the energy, the size, the fitness. All of these things are what we're going to try to write down in a mathematical language so that we can kind of sit where the organism sits. In modern machine learning, we call all of these deep neural networks black boxes. So information goes in and some behavior of that algorithm comes out. I would argue, of course, that these aren't black boxes at all. They're just hard to interpret because they look like, say, this. This is like ResNet. This is just a sketch of a piece of ResNet. That's just a high-level diagram of what this incredibly wonderful, but yet now old and dusty neural network does for you. And of course, some of you are old enough to recognize this Commodore 64 board. In both of these cases, we can interrogate all of the pieces of this circuitry, but it doesn't give us a lot of insight about what the goal of computing is. And if we sat with these circuits without any knowledge of their inputs and outputs, we would have a hard time inferring what they're doing. Now, thankfully, biology gives us this kind of more what I've called a blue box. So what's in the blue box are not those kind of circuits. They're molecules, they're membranes, they're whole cells, they're groups of cells, they're synapses here. I've got neurons for us all here, or they're entire organisms or groups of organisms like fly, fish, and this macaque. So when we look inside these systems, it's often easier to guess at what they're doing by looking at structure and by looking at their inputs and outputs. But they're not generalized computation devices. And that actually helps us for understanding and building intuition. So what we want to do today is to understand how we constrain one of these blue box systems to perform a certain behavior, and how we can like recapitulate that in silico or interrogate it with information theoretic tools that we borrow from machine learning to understand trade-offs. And so here I'm going to write for you a general information bottleneck plane. So here on this x-axis, I have throwing away or compression. Imagine that this is what the organism does with its inputs. So this is more and more throwing away until you have nothing. And of course, if you have nothing, you can't do what matters. So here we're going to talk about prediction as one guess at what matters to an organism, but it could be fitness or anything else you'd like to put on this y-axis. That's the beauty of some of the techniques we're going to talk about today, is that you as the theorist can put whatever you like in, and that gives you a way of testing your hypothesis of whether or not that's what the biological system cares about. Of course, as you throw away more and more and more, you start to lose what matters until you have nothing. Here, this slope is always less than 1 no matter where you are. So there's a forbidden region where you can't get more out than what you put in, which is just sort of think of it as a data processing inequality. And because there is noise in the external world, noise in neural firing, noise in external behavior, there is an asymptote that you reach. So there's a maximal amount you can know about whatever it is you think that matters as you encode more and more from the external world. All right. So we'd like to know where biological systems sit. So as theorists, we're going to be able to compute this bound at the edge of, the forbidden region. This is what we would say is optimal. And if we're allowed to play with the throwing away and compression, we might want to sit in this elbow where we've not yet encountered the land of diminishing returns. Or if we don't have access to modifying this, it's just what the brain gives us, we'll find a point in this space and then ask if the biological system hits our bound that we've calculated. So this is what we want to know. We want to know where the blue box sits in this plane that's allowed. So as David said, my group has really focused on prediction and has asked questions about the prediction in the retina. And this is reviewing both older work that kind of kicked off our whole foray into these things from the end of my postdoc to the beginning of our group's work, and then is now coming around full circle, where we're coming back to this question of prediction in the retina and asking mechanistically how that happens. And Jared Salisbury and Kyle Boyanek are the two folks whose work I'll highlight a little bit, who are actively doing this right now in the lab. And some folks that I highlighted at the beginning have graduated and used what they've learned to move on in their own careers. So as we all know, the retina is a piece of the central brain that happens to be sitting in your eye. Very convenient for us as experimentalists that there's this piece of the periphery slammed up against central brain circuitry in this accessible and kind of extractable part of the brain. There aren't that many centrifugal projections to the retina, at least not ones that we sort of decide to not care about them, and we cross our fingers and hope they're not important. But for the most part, there isn't a lot of, say, cortical feedback or any cortical feedback to the retina to modify how it's processing. So if we can record from the retina, we can see what the brain sees and we can see what that input-output transformation is. And the retina does a lot. So if we take a slice through the retina, there's this complex circuitry. Here's your input photoreceptor layer. Here's your output retinal ganglion cell layer. And this is from Vreeswijk's review that shows that even if you take the simplest cut of what the retinal circuitry is like, it's got different cell types and it's already complicated. Just parsimony arguments would tell you that you don't build something like this to report a photoreceptor by photoreceptor picture of what the world is doing. And of course, if we look at some of the work from Thomas Euler's group, even just looking at retinal ganglion cell types here, we can, whether you're a lumper or a splitter, there are lots of different functional classes. And of course, this is too small for you to interrogate in real time. But what this is supposed to highlight to you, it gives you the impression that there are many, many different functional classes that they are pulling apart with these different stimuli here in the mouse retina. Okay, so all of this says that the retina is a complex piece of neural circuitry. And it, over the years, we have discovered as a community many, many, many complicated nonlinear processing features that make the retina a good kind of computer. So here I'm highlighting a rapid fire introduction to non-textbook-like behavior. Pardon me for 1 second. In the retina. Going back to early work from Meister and Barry when he was a postdoc in the Meister lab, showing that the retina does a good job of motion anticipation. The neural image of a bar on the retina, so this is position on the retina. When you flash the bar, looks like this, but when you drag the bar in, when it's moving, the retinal image skews towards the leading edge of the bar's motion, which are all things, all of these here that are highlighted are things that can contribute to something that feels like a prediction in the brain. And what we set out to do in my group was to ask whether or not there's a general way of quantifying how well the retina does prediction as a whole. And to do that, instead of saying, oh, if there's smooth motion, does it predict? Oh, if the motion reverses, does it signal some sort of surprise? Omitted stimulus response that Greg Schwartz worked on when he was a grad student in the Barry lab shows you some other cool nonlinear features of retinal ganglion cells. And of course, Benseovetsky's key work showed that retinal ganglion cells have object motion sensitivity. So we want to take this kind of holistic view about prediction in the retina. And to do that, what we aimed for was to present the retina with both predictable and non-predictable components of motion and ask if this is the predictable part and this is the not predictable part. As the retina compresses, does it preferentially kill or throw away the non-predictable parts of motion? So to do that, we needed a moving bar that would have predictable and non-predictable components to the motion. So we just made a stochastically driven damped harmonic oscillator that you can see an example of here. So it's tethered to the center of the screen by a fictive spring, and there's some inertia to the bar. So from high school or freshman physics, we know how to write down the equation for a damped harmonic oscillator. And this is one that, let me get that off, that just happens to be driven by gamma of t is a white noise process with an effective diffusion constant that's parameterized by d. So these components of the motion are predictable. We all learned how to solve this equation with initial conditions. We can tell you what's going to happen to the bar for infinite time afterwards. This part adds the non-predictable complexity to it. And we want to know whether or not the retina, when it views this kind of motion, throws away the parts that keep track of, say, the whole trace of the stochastic kicks that the bar got. So, sorry, just I will try not to pay attention to the chat for a little while, but I see questions in there, so don't worry, I won't forget you. So when we record from the retina, we put the retina from either a salamander or a rat on a glass slide with a multi-electrode array embedded in it. And it is one of the beautiful things about modern neuroscience that we can record with high fidelity from complete neural populations. What we're going to do is summarize all of that as ones and zeros, as whether or not in a small time window of about 20 milliseconds, the spikes from that collection of neurons in the retina either spiked and we give that a 1, or not spiked, we give it a 0. And what we want to know is how the retina compresses that 1D trace of the bar motion from the past into the spiking at present, and then how much that spiking at present gives information about what's happening in the future. So this defines a classic information bottleneck problem where the relevant variable of what we're trying to maintain, what we say matters to the organism, is the future, is how much those retinal ganglion cells have to say about the stimulus S in the future. So this embodies, we get to put, when we do an information bottleneck, when we trade off one thing from the other, when we trade off compression with what's relevant, we get to say what's relevant. And we have said the future is what's relevant. So we're making a hypothesis that the retina might care about prediction, and we're going to be able to quantify exactly how much would be optimal in this case. So that's the equation of motion we have there that we've defined, that we put in. And of course, this ability to predict depends on the correlation structure of that motion that we put in. So when we do that, this equation is solvable. It obeys, it's a jointly Gaussian input statistics. So past and future are defined by Gaussian. So we can solve this exactly. And we follow the work that came before us from Gal Chechik and Tali Tishby and others about how to do that. We go in, here are some references if you would like them. We solve this sort of summarized information bottleneck, and we can solve for this bound. So how much any system knows about the past in this bar movie, how much we can say how much it will know about the future, that defines this bound. And we want to ask, again, where does the retina sit in this information plane? And what we found is that the retina gives us access, it compresses about this much in small groups of cells where we can measure these quantities faithfully. And we put that together and we find that the retina does appear, the salamander retina here, to saturate the bound. So we compute the information the retina has about the past and the information the retina has about the future, and it sits close to the bound. And what I mean by close is that if you look at the size of these error bars, these are both error bars in the variation across groups of cells that we grab from our recording, and also our confidence, our ability to measure these information quantities. So I would say that here we're given a discriminability of like not caring about the future, sort of medium and high, and that's about it. So we get some categories here. But we do find that the retina saturates this bound for the salamander at a 20 millisecond, 16.7 millisecond lag into the future. We find that for the salamander, and we find similar things in collaboration with Olivier Marr, where one of my students, two of my students have had Chateaubriand fellowships to go collect data in Paris, lucky students, have also shown that this works in the rat. So we have motivated this way of looking at what's happening in the retina by prediction in the retina. I'm sorry, I'm at my parents' house and the phone is ringing. Hopefully that's not too loud. And we see that there is a kind of saturation of the bound. So what we'd like to ask, to come back to this notion of embracing the biological complexity, is how prediction operates in complex natural behaviors. And this is a place where we're going to need modern machine learning tools to make progress. So here's the natural behavior we're going to look at. We're going to look at a fly making an escape behavior. So here's the fly. This is slowed down about 400 times. Here's the dragonfly that wants to eat it. And here is the fly's banked and then counterbanked escape turn. These are beautiful, beautiful behavioral data collected by Michael Dickinson's group and available online. So we can actually use this kind of complex natural behavior to ask about optimal prediction. And to do that, we need to, again, look at what the fly actually does and how it's going to predict how to control its wings during this escape maneuver, because it doesn't have time to wait for feedback. So let me show you, give you a sense of the numbers. Here's the fly's banked and counterbanked turn. That whole thing lasts about 40 milliseconds. 40 milliseconds is super fast, even for the fly. Now, their whole visual processing system is quite speedy, but there is a gap. Without some sort of feed-forward visual prediction helping steer this behavior, there would be a 20 millisecond sort of no known control to the system, because the haltear feedback, the little gyroscopes the second wings are modified into on these flies, doesn't come in for 20 milliseconds. And this turn is a 10x faster angular velocity than what the fly does when it's regularly flying around. The fly needs to be able to control carefully that escape. It can't just play a motor tape, because it has to know where it's initiated that turn and how its body is oriented in space, as well as the direction it wants to head in. So our hypothesis was that feed-forward visual prediction contributes to ego rotation control during this first kind of open loop gap where there isn't active feedback control available to the fly. So what we did here was we took the fly's natural motion. So we took those from the Michael Dickinson group that are available online. That's our first step. Then we painted an external visual world with natural scene statistics from the Van Hatteren database and played it to a model of the fly vertical sensing system, because when you're making a banked and counterbanked turn, it's the vertical motion that matters. So we're going to play the motion from one into a visual system that is experiencing that ego rotation with natural scene statistics painted on the background. Next, we're going to look at how the input and output responses of the vertical sensing system. And for this, we have a really good validated over decades of research fly wiring diagram for the vertical sensing system from our colleagues, Axel Borscht and Idan Segev. And then we want to solve this bottleneck problem. We want to ask, how does the information from what the fly experienced in the past give it information about how it needs to control its how it can predict what its vertical sensing system is going to experience so that it can control its wings in the future. And this is wickedly unsolvable. We've got a natural behavior in a natural environment played through a complex neural circuit that thankfully we can model. We don't have to go measure this in the fly, but it'd be lovely if somebody would like to do that. We have absolutely no hope of solving this analytically. So this is where machine learning comes in to the rescue. So again, we want to be able to compute this bound. And here we did compute this bound. And what we did was use open source software called Milka and to obtain mutual information estimates. And it's kind of like a k-nearest neighbor approach. And the mutual information is approximated via its future from the past. It's using some sort of corresponding like gamma function approximation. And that lets us get access to this bound and then put on top of it what the fly's vertical sensing system does. So again, this is that blue box where the neural system lives. It can live anywhere in this plane. And to predict 10 milliseconds into the future, the output of the entire vertical sensing system sits right at the bound and close almost to this elbow where it would have diminishing returns if it kept more past info. Now, we were particularly interested in triplets of outputs from the system because we know something about the anatomy and where everything projects. And what Sway could do, which was a huge advantage in the system, was that she could look at this model of the fly vertical sensing system, its inputs and outputs, and sort of, kind of like pulling apart the fly. She could pull apart the fly's brain systematically. And what she found was that the key piece of this system that she could not mess with without messing up prediction were these axonal gap junctions between the vertical sensing cells. And when she X'd those out, saturating the bound fell away. The vertical sensing system, so this system was constrained to have the same past info, didn't have the same future info. The triplets, it screwed up everything. And they slid down this bound almost all the way to the place where they don't have enough information to give a viable prediction. So we were really encouraged by this because we thought, oh, great, we're able to compute this bound even though the problem is super hairy. We can put the biology on this bound using the real behavior, what we know about natural scene statistics, and what we know about how to create an in silico fly eye. But we wanted to know more about what is being computed here. That's often a critique of using information theory measures, is that I tell you there's, so many bits, and you could very reasonably say, so what? Why should I care about so many bits? And I'd say, well, it's close to the bound. You say, well, what is that? Does this make sense? Can I have some more intuition about what's going into this? So to take that next step further, we used what are called variational information bottleneck techniques, again, from machine learning. There was a huge surge of papers on so-called VIB methods in the literature a few years ago. And we could use those to look at the latent space of a network that approximates this bound and see what that looks like. So a variational information bottleneck technique is basically like a predictive autoencoder. So there's an input and an output, and we say the input is the past and the output is the future. But what it does is compress into this latent set of variables, and we can look at the structure of those latent variables. This is a general picture of what these VIB techniques look like from this nice review, if you want to know more. But back to the retina, but back to the fly, what we then do is just do simple PCA on those VIB latent spaces. And then we map into that latent space different heading directions of the fly, because we were wondering, like, maybe what this does is give the fly a sense of what direction it's about to do this banked and counterbanked turn, because that's what it needs to do for control. And to have that, it needs to know where it is in space, not just where it's heading, but where it is in space and how those things combine. And so here you see our inputs of different heading directions in just this little tiny quadrant of the fly's visual system. So 19 degrees, 37 degrees. And what I hope you can see is that they project onto discriminable parts of this plane. And if we look at these points after they run through the fly visual system, so we look at different stages in this latent space, defining different stages of moving through the fly VS system, we find that it goes from this, which is discriminable, to something that's along a line that's discriminable with a simple linear readout. So what machine learning tools let us do is go all in and embrace the complexity of real neural systems and embrace the complexity of real neural behavior, and still solve those tricky, thorny processing steps to understand what we think might be happening in the retina. Okay, so the fly visual system in the fly. I said I'm already into I'm predicting too far ahead in my slides myself. So I want to take you back to the retina and talk about some recent work from Ben and Caroline that's on bioRxiv now. And I want to tell you about this because, again, we come back to things where we can't really compute anything. So let me tell you a little bit about what we did, and then I'm going to bring it around to prediction again. So first, let me motivate this. So take a deep breath. We're leaving the fly behind. We're coming back to the retina of the salamander. So, and we're going to look at how the salamander responds to all these different kinds of motion. So these are scenes from our motion database that's available through some Globus download. If you have any trouble with it, ping me, because sometimes the system is a little wonky. But it's 2 terabytes, so you need some server to help you download this. These are natural motion videos that we collected in the field with cameras that have optics that match different kinds of eyes. So this is like a larval salamander buffet movie, a fish swimming around a tank. There are optic flow movies, there are water movies, there are lots of foliage blowing in the breeze movies. And what we did in collaboration with Olivier and Michael, so Mar and Barry, was play these movies in a sort of pseudo-random order over and over again to the salamander retina. And we wanted to make a model of what the vocabulary of the retinal response was. So we're going to kind of make a classical Maxent model of the population response. And what I mean by that is if we, again, bin time and think about the population response as a binary vector of ones and zeros across cells, we're going to say that the probability of observing any particular pattern of firing across those cells has some probability that defines something from an exponential family. So it's a kind of energy-based model. And it has this look like an Ising model if you're a physicist. There are local fields on the neurons which define kind of their excitability or their, sort of related to their firing rate, and coupling terms between the cells. And what Ben and Caroline wanted to do was to put a time dependence on those fields, but not on the correlations. So what we're imagining is that we're scooping up all of this kind of stimulus-driven time by time point complexity into these local fields for every neuron. And we're going to see how their correlation structure matters, what it matters and how it changes scene to scene. This is just to say that this model does well. So these on a log-log plot are the probability of all of the spiking patterns that we observe. And here's how the model does. And this is unity. And apart from the ones that the patterns we only see once or twice in the whole recording, we do pretty well recapitulating what we see in the retina. And this is following on lots of previous work from folks like Elad Schneiderman, Gasper Tkachuk, Bill Bialik, and others. We want to look at the part that isn't the moment by moment driven by the external stimulus. So we're going to look at this JIJ, this coupling matrix. I'm going to flash up a whole bunch of coupling matrices for you. So be prepared. This is a number of cells by number of cells, and how strong is that interaction? For the tree movie, where the tree is blowing around in the breeze, this is cells by cells. And I'm showing you just a few cells here from the whole population. And what I hope you can see is that there's a pattern of some strong connections against a background of fairly weak sort of interactions, and not connections. This is the retina, so nothing is sort of directly connected. But when we look at the grasses movie, this is that same matrix for the same group of cells. And what I hope, if you squint at this, you can be convinced that the pattern here is similar to the pattern there. There's kind of like a backbone of sparse interactions that remain the same. They have different values, but the skeleton remains the same across movies. And that's true no matter what we did. And we also did this for the checkerboard movie, where we can't make the stimulus-dependent Ising model, this time-dependent Ising model, but we can use direct coupling analysis to get a matrix like that. And it has similar structure too. So our question was, how much can we throw away here? What do these interactions do? What can we throw away? So here's a distribution of those interaction weights across a population of cells. And you see that this is on a log plot. And we have a lot of small, we have a lot of small connections, and then a heavy tail of strong connections. What we wanted to know is if we could chop this off and just use those strong connections, and would we get the same sort of behavior? Would we be able to still explain what the retina is saying to us when it spikes? And the answer to that is yes. If we don't include the couplings, we get the blue fuzz along the unity line. And again, we want the sparse model to match the full model. And if we only take the top 10% of couplings, which here, because there's about 100 cells, scales like n, the number of neurons, we get pretty close. So we can shave away quite a lot of this and recapitulate what the brain seems to be saying to us in terms of its spiking vocabulary. Now, this doesn't, again, we want to come back to what this could mean, because this is something that stays the same scene to scene. But the values that are like kind of projected onto that interaction space change. So what we found is that these interactions are critical for deciding what scene you're in. So if the salamander changes location, as they do in their life, from a sort of aquatic scene to a leafy scene when they're in their adult form, they need to know sort of how to compute and what's going on when they move from scene to scene. And this is just a Bayesian approach asking, what if I'm just observing spikes from the retina, which sort of natural scene natural movie am I in? And this shows you how, as the sort of a Bayesian estimator accumulates information by observing spikes from the retina, it quickly learns to correctly identify which scene you're in. So this means that your scene ID sampling across movies and across samples comes up and gives you a good estimate of what scene you're in after just a second or two, which is a good thing, because the first thing that happens when you move from scene to scene is that your retina does a lot of sort of contrast normalization and adjustment. So there's a lot of adaptation that comes in, but what you retain is an estimate of where you are. So you don't adapt everything away. And if we shave this down to just the backbone, we get the exact same kind of behavior out. But if we get rid of those interactions, those sparse 10% of interactions, we get the blue, and it takes many, many, many more seconds to know where you are. Okay, I have a last short piece. This is on the same data. So what we think is that those interactions are important for knowing what scene you're in, but we want to come back to those time-dependent fields. So what we're asking here is, when we're in this rich natural environment, do we throw away different things for different tasks? And this is work from Soe. And what she did was, again, embrace modern machine learning. And she looked at a UNet. And what she did was feed the UNet, which is, again, a kind of, the way she used it as a predictive autoencoder. So she put in independent PSTHs, so the firing rates of individual cells, ignoring all of the couplings, no couplings here. And she made an encoder decoder with a highly reduced dimension latent space Z to predict what the future scene was going to be, a frame. So this encoder decoder is taking in time-varying firing rates from the whole retinal population in the past and trying to predict what pixel by pixel frame. So it's going to learn a lot of things that are specific to what it's trained on, but we wanted to know how it would generalize. So what we did was to train this encoder decoder on responses from, say, the fish movie, freeze the encoder. So here we're freezing it on the fish. So that's why there are frozen fish sticks here for you to guide your intuition about this. So this encoder is always frozen on the fish movie, but we want to give it other movies like the water or the leaves and see if it has the same kind of latent space, and if that latent space then can predict what the future frame will be. And remarkably, the performance on predicting where you are in a natural movie, as you increase the dimension of Z, again, using tools from modern machine learning to play with these neural networks, artificial neural networks, to understand the structure of the space. You increase the dimensionality, you saturate on performance in saying where you are and where you're going to be in this movie. And if we swap out and freeze the latent space on a particular scene, we get some generalization across scenes. So if we take the fish and we freeze the latent space on the fish, but then we look at leaf inputs, we get the same predictability about what scene you're in, about what part of the movie you're in, about the particulars about where you sit there. So I was going to try to tell you two more things, and I'm not going to, and I'm not even going to summarize them, because that's not fair. I know sometimes I have this instinct to just say, let me give you a 5-minute summary of things that are way too much to take in. But instead, I'm gonna wrap here and say that the overall picture of what I wanted to tell you is that the sort of horror of biology that scares all of us who want to, be able to compute things and gain intuition and make theories about it, that complexity, that you can allay those fears. You can embrace complexity of natural stimuli and natural behavior by asking questions where you make hypotheses about what matters and then interrogating them using this plethora of tools that we have from modern machine learning. And I think that's really a benefit of where we sit now in modern neuroscience, that we have beautiful behavioral recordings, nearly complete neural connectome data, neural recordings that we can use, and we can gain understanding and intuition about, again, how these blue boxes work by using these modern tools. So with that, I'm going to leave you with some references and hopefully haven't completely overwhelmed you today, and I'd be delighted to take your questions. Thank you so much.